The haptoglobin located in the 16th chromosome, more known as the HP gene, is produced in the liver, and it joins to the free hemoglobin, forming hemoglobin-haptoglobin complexes removed from the circulation through the liver and catabolized by the liver parenchyma cells. The parenchyma liver cells are a functional tissue of the organ that make up the 18% of the liver volume as hepatocytes. So the hemoglobin accumulated in the kidney is secreted in the urine. An important fact is that haptoglobin acts as an antioxidant. The principal function of hemoglobin is to collect oxygen from the pulmonary alveoli and communicate it to the tissues, take carbon dioxide from them and transport them back to the lungs to expel them. The haptoglobin belongs to the group of peptidases S1, but before being processed and cut, the uncut form forms zonulin protein. Zonulin is a protein that modulates the permeability of junctions between cells and the wall of the digestive tract. The principal function of zonulin is to regulate the flow of molecules between the intestine and the bloodstream, allowing the passage of nutrients and blocking the entrance of macromolecules. There are two factors that cause the release of zonulin: intestinal bacteria and gluten. The structure of haptoglobin consists of two alpha and two beta chains connected by a disulfide bridge. A disulfide bridge is a functional group. It is also called a double S bond. In biology, this concept refers to an important component of the secondary and tertiary structure of proteins. The chains that form the structure of haptoglobin are originated from a precursor protein, also called a proprotein or propeptide. It's an inactive protein that can be converted into an active form by a post-translational modification, like breaking a part of the molecule or adding another one. A post-translational modification refers to a covalent or enzymatic modification of proteins. So, before being processed, the haptoglobin had 406 amino acids. But as the final protein contains two alpha chains and two beta chains, it has 776 amino acids. The haptoglobin is produced by hepatic cells. The hepatic cells are also known as perisinusoidal cells or ethel cells, are found in the liver, more known as the space of Dice, a small area between the sinusoids. This word refers to a sinusoidal capillary, and a capillary is a small blood vessel that has a length of 5 to 10 micrometers. So, there is between sinusoids and hepatocytes. An hepatocyte is a cell that belongs to the parenchymal tissue of the liver. An interesting fact is that the hepatocyte made up the 55 to 65% of the liver mass. These cells are involved in protein synthesis, protein storage, and the transformation of carbohydrates. They have a polyhedral shape. They usually have one or two nucleus, multiple organelles, and their cytoplasm contains glycogen that is mainly stored in the liver and muscle, provides energy to the body, and fat. They are very active cells that are constantly forming new proteins, for example, albumin. If this gene doesn't work or the haptoglobin is lower than the normal levels, a disease can be developed. For example, hemolytic anemia, prolonged liver diseases, accumulation of blood under skin, like varices, or an hepatic disease. An increase in haptoglobin means inflammation, neoplasm, myocardial infarction, and Hodgkin disease. I'm gonna explain some diseases. Hodgkin disease is a type of lymphoma. A lymphoma is a cancerous part of the immune system. This disease can spread to nearby lymph nodes. Some symptoms are weight loss, itchy skin, fever, and loss of appetite. Hepatic disease, more known as liver disease, is a type of damage or problem in our liver. Some symptoms are jaundice, more known as icterus, is a yellowish pigmentation that manifests in the skin due to the high bilirubin levels, accumulation of fluid in the abdominal cavity, risk of bleeding symptoms that take place in the gastrointestinal